story begins with 6 unlikely heroes: the DNA template strand, the RNA polymerase strand, cytosine, guanine, uracil, and adenine. I live in the nucleus envelope, but I've always wanted to explore the cell, but I'm too big. But my hope is for my dreams to go for me. Don't worry, I'll help you get there. In order for a part of the DNA to go out of the cell, it must first be transcribed into RNA, and it all starts with initiation. Don't worry, DNA template strand. Because I'm on the job, your kids will be out of the cell making proteins in no time. I just have to start. Hi, I'm promoter, and you need to start transcription right here. And on this side of the 3' 5' strand, and you have to go that way. That seems kind of tricky. Don't worry, I'll be there the entire way to help you. The promoter will help the RNA polymerase on what genes get expressed. The RNA polymerase strand starts to then unwind the DNA template strand from the coding strand, and in comes the 4 unlikely heroes. Okay, the DNA is unwound, and this is cytosine. I go by CTP, actually. Oh, okay, that's cool. You just have to bind together the nucleotides for DNA. It's adenine to thymine and guanine to cytosine. Okay, so this is cytosine, so guanine goes here. And this is adenine, so thymine goes here. For RNA, adenine doesn't bind to thymine. That's only for DNA. For RNA, adenine binds with uracil. After the RNA polymerase unwinds the DNA template strand from the coding strand and transcribes the template strand into RNA, and rewinding the coding strand and the template strand, RNA polymerase has now reached its stop. RNA polymerase, your job is done here. You have made an mRNA that is now able to exit the nucleus. This specific DNA sequence proteins are going to help you now. We're going to help you unbind from the DNA and mRNA to exit the ribosomes, to go to the ribosomes. Don't worry, I'm gonna miss you, DNA polymerase. All right, don't worry.
As mRNA leaves the nucleus and enters the cytoplasm, it's now time for translation. Translation is the process of translating the nucleotide sequence on an mRNA strand into an amino acid sequence. The amino acids are bonded, forming a polypeptide chain, aka a protein. mRNA is made up of codons. These are sequences of 3 nucleotide bases that code for one specific amino acid. tRNA's job is to read the mRNA codons and deliver the right amino acid. tRNA has an anticodon, which is a triplet of mRNA bases that are complementary to an mRNA codon for a specific amino acid. This is where tRNA bonds to mRNA site. Translation takes place at the ribosomes and occurs in 3 steps similar to transcription, including initiation, elongation, and termination. Step 1 of translation is initiation. Hold up, I think I'm lost. I'm the small subunit of the ribosome. To start initiation, I bind to a recognition sequence on the mRNA strand. As seen here, this is the small ribosomal subunit, the mRNA strand, and this is the tRNA charged with methionine and bonded to the mRNA strand. Here comes the large subunit of the ribosome, the final ingredient for the initiation complex. The second step of translation is elongation. As seen here, the first tRNA is in the P site and it is bonded to a codon on the mRNA strand. But here comes another tRNA molecule. During elongation, the large ribosomal subunit catalyzes 2 reactions. First, it breaks the bond between the tRNA in the P site and its amino acid. Then it forms a polypeptide bond between the first amino acid and the amino acid that is on the tRNA currently in the A site. This tRNA is entering the A site in this diagram. The tRNA now has a growing polypeptide tail. The final step of translation is termination. Newly completed polypeptide separates from the ribosome and it is now a complete protein.